Dear students, I welcome you all to the 12th session of Reproductive Biology and Developmental Biology paper prescribed by the Mangalore University for 6th semester BSc Zoology. In this session, we shall look into some theories, classical theories that were put forth by some scientists who extensively worked in this field. So we have to look into many theories in this chapter, theories of development. The first theory we are going to study in this chapter is the germplasm theory of Weismann. The germplasm theory was proposed by August Weismann. The gentleman whom you can see in this photograph is August Weismann. In 1883, and can be considered as a landmark theory in developmental biology because he was the first man to distinguish the germplasm as a separate entity from the somatoplasm. That means he was the first man to describe the two different cytoplasms are present in the living system, the germplasm and the somatoplasm. The somatoplasm is the one that produces the entire body, and the germplasm is responsible for the continuation of the generation. According to him, the germplasm is a self-perpetuating cellular entity which continues its existence through ages. The child or the next generation inherits characters of the parents through the sex cells of the parents and not from other parts of the body. The germ cells acquire the characters from pre-existing germ cells. In each new generation, a temporary soma or body is built around the germplasm that descended from the parents. According to Weismann, the embryo remains already organized in the chromosomes of the nucleus. And during early development, these characters are unpacked in an orderly manner. So that means he was the first man to believe that there is something called chromosome, though he did not know anything about the importance of chromosome in inheritance factors. He considered these chromosomes as determinants those days. And he thought that these embryos remains already organized in these determinants that are present inside the nucleus, and just during the developmental process it will simply rejuvenate. He recognized the units of heredity as determinants, and these are segregated during the early cell divisions of the embryo, making these divisions differentials. In the first cell division, the determinants are distributed into right and left sides of the future embryo, and later divisions into anterior and posterior parts and so on. Later interpretation between them enable the epigenetic development possible. See, most of the postulates of this theory may look weird these days because we know many more things about development and genetics and the epigenetic influence of the epigenetic influence on the process of development, everything. But those days they never knew anything about these things. So probably they believe that these determinants are the key reasons for development. After the theory put forth by Weismann, the next gentleman about whom we have studied in detail in the 11th session while studying about the history of developmental biology, Wilhelm Raux proposed one more theory called mosaic theory of development. In 1888, Wilhelm Raux performed an experiment on frog's egg. He kills the nucleus of frog's blastula at two-cell stage by touching one of the two blastomeres with a red-hot needle. You can see here this fertilized frog egg, and the needle is inserted into one of the blastomere immediately after the first cleavage, so that the nucleus in this cell is destroyed. So what happened later on when the developmental processes proceeded? You can see that part of the embryo which is inserted with a red-hot needle. That means it was killed or it was destructed. The physiology of the nucleus of this particular part is disturbed. Hence the development is arrested here. No development here. Only the part of the section of blastula is developing. He later observed that the embryo developed with 1/2 part defective. Thus it was concluded that certain areas of egg were predetermined to develop into specific part of the body. See here, only one part of the body which was unaffected developed completely. The other, remains as such. So this gave him some hints about, the predetermination of a particular part about the development. So this part may become something in the future. So if you kill them in the early embryonic stages, then the future parts which are destined to become by this particular region may not be formed in the later stages. The animal pole, as you know that the ovum or the egg has two poles, the animal pole and the vegetal pole. The animal pole is more actively dividing, and it will involve in the process of development more actively than the vegetal pole, because the yolk will be concentrated more towards the vegetal pole. So the animal pole is predetermined to develop into anterior side of the embryo, and the vegetal pole into the posterior part. The gray crescent develops into the blastopore. See here, we can see here the animal pole and the vegetal pole are present, and a gray crescent is formed, and this gray crescent is formed in the fertilized egg of the amphibians. This is due to the movement of the pigmented cell, pigmented cytoplasm after the fertilization. So in between animal pole and vegetal pole, near the place where the blastopore is going to develop, a gray crescent is formed. During cleavages, the predestined regions, predestined means ಮೊದಲೇ ಅದರ ಭವಿಷ್ಯ ತೀರ್ಮಾನ ಆಗಿದೆ ಅಂತ, ಮುಂದೇನಾಗಬೇಕು, are segregated into the blastomeres that develop into specific tissues and organs in the future. The blastula thus is a mosaic of blastomeres with potencies to develop into different parts of the body. And as development proceeds, these are differentiated and segregate into specific cells. So you can see here, once the blastula is formed, after blastula, the next stage, the gastrula, will be formed due to the movements of the cells here, which will produce an ectoderm, endoderm and a mesoderm as well. The next theory of development proposed was a regulative theory, which was proposed by Drich in 1891. This is a result of his experiments with the sea urchin eggs. The regulative theory is in contradiction to the mosaic theory of rocks. Hans Drich was a German scientist, observed that the blastomeres of a sea urchin at two-cell stage, when separated from each other, grow to develop into two young embryos of comparatively smaller sizes. So you have a zygote here, and in two-cell stage, if you separate the two cells and allow them to grow, they will become the larva and the embryo larva and then the adult. But if you separate the two cells, then both the cells will develop into embryo, larva and adults, but comparatively smaller, a little smaller than the normal larva that it would have produced if it was in intact position. At 4-cell stage, he continued the experiment. At 4-cell stage, the separation of blastomeres led to the formation of 4 distinct embryos, you can see here, but with even small in size. So you can see here, if all the 4 cells of a zygote after a second cleavage was intact, and a normal larva would have been developed, which is larger in size, but if you separate these 4 cells, each 4 cell has the potency to develop into a complete embryo and the larva later on, but only the difference is the size. So these will be becoming smaller in size rather than when you compare it with the normal larva developed from the intact zygote. So if intact, all 4 blastomeres formed only one embryo. Okay, this is a fascinating thing. If you separate them, they become 4 embryos. If they are intact, then only one embryo. No 4 embryos are produced. However, this pattern is continued only for few cleavages. Later cleavages could not produce separate fully formed embryos upon isolation of the blastomeres. So this potency of the cell will be retained only up to 8-cell stage or 16-cell stage, not more than that. So once if the cleavage proceeds this stage, then the cells which are isolated may not produce fully formed embryos. Instead, they may produce part of the embryo, which in the future they are going to produce. Therefore, Drich concluded that the early cleavages of egg were all equal. So initially they were all equal. The homogeneous material of the ovum was quantitatively equally distributed into the blastomeres. So the conclusion was initial cleavages or the initial mitotic divisions were completely equal, including the quantitative separation, quantitative distribution of the cytoplasm. That means if the cytoplasm, quantity of the cytoplasm is 4 parts in a single zygote, then it will be divided into 4 parts, and each part is offered to each daughter cell. So this is quantitative distribution. In early stages, all the blastomeres have equal potencies to develop into whole embryo. So this is the conclusion of the theory proposed by Drich. However, if not separated, if their fate in the whole is determined by the position occupied them in whole blastula. See, this is one of the curious things you are supposed to see. If you separate them, they will become new individuals. If you don't separate, then they will occupy different positions. They will not die, right? They will occupy different positions and they become the different parts of the future embryos. So this type of development is known as regulative development. Each cleaving egg, according to Drich, was a harmonious and equipotent entity. Harmonious and equipotent entity. Each blastomere has potentially the properties of the whole. At present, it is known that every egg is both regulative and mosaic in development, but those days they never knew that, so different theories were put forth. The extent of regulation is dependent upon the mRNA content of the cytoplasm. So the quantity of the extent of regulation is dependent upon the mRNAs that are synthesized and packed in the ovum during oogenesis. Okay, the quantity of that mRNA determines whether the cell must be going for mosaic development or for regulative development. The next theory is the gradient theory of child. The experiments conducted by Theodore Boveri, Child, Horstadius, Ronstrom and Jefferson on sea urchin eggs have given a new concept to the experimental biology or embryology. Boveri in 1901 noted that the vegetal pole region of the sea urchin egg controlled the activities of the regions situated towards the animal pole. In fact, the vegetal pole region had dominant effect over the animal pole areas. No reason was given by him, but Child in 1940 proposed the metabolic axial gradient theory. So you can see here there are two poles, the animal pole and vegetal pole. Initially, through their experiments on sea urchin, they found that the vegetal pole controlled the activities of the regions that are situated in the animal pole. So this vegetal pole has the impact on the parts and cells of the animal pole. So this was the theory they put forth. So according to this theory, the single physiological gradient, namely the metabolic axial gradient, controlled the process of morphogenesis during the development of sea urchin. So the reason behind this conclusion of the metabolic axial gradient was, Child observed that there was a vast difference in the rate of general oxidative metabolism in the eggs of sea urchin between the vegetal pole and the animal pole. If you look at the rate of metabolism at vegetal pole and animal pole, it is completely different. Though it is believed to be similar in earlier days, experimentally, if you look at the rate of metabolism into these two different poles, it's quite different. So the rate of metabolism was quite higher, quite greater in animal pole region, and it gradually decreased towards the vegetal pole in a gradient fashion, a graded fashion. Okay, similar gradient existed in developing eggs of the starfish as well. So he also measured the rate of metabolism in starfish egg also. There also he got the same result. The animal pole region, being more active metabolically, divides at higher frequency as compared to less active vegetal pole region. The reason we know right now is because of the accumulation of yolk, but they did not know earlier. So they believe that the animal pole is more active, it is producing more metabolic oxidative reactions. Hence, this will divide more rapidly than the vegetal pole. Hence, the cells of the animal pole will be relatively smaller than the vegetal pole, because vegetal pole is dividing bit slowly than the cells of the animal pole. Thus, a metabolic gradient is found along the animal-vegetal axis, with high metabolic rate in animal pole region and gradually decreasing toward the vegetal pole. So, if you plot this graphically, the rate of metabolic activity from animal poles towards the vegetal pole, then you can see there is a decrease in the activities from the animal pole towards the vegetal pole. Hence, a animal-vegetal axis of metabolic gradient can be observed, and this is gradually decreasing towards the vegetal pole. So, this Child's original concept of metabolic gradient has been modified later years by many scientists like Horstedius and Runstrom. These biologists observed a double gradient system extending along the animal-vegetal axis. These are the animal gradient and vegetal gradient. The animal gradient is most active at the animal pole region and gradually decreases in its intensity towards the vegetal pole, and the reverse is the vegetal gradient, on the other hand, is most active at vegetal pole region and decreases towards the animal pole region. See, the Child's belief was, Child was a scientist, okay. His belief was there is a gradient. He believed in a single gradient, that is animal-vegetal axis gradient of metabolic activities, where he believed that metabolic activities are more vigorous in animal pole and it is constantly decreasing, gradually decreasing towards the vegetal pole. But later on, through the experiments, the metabolic gradient theory was modified. They modified it by saying there are two gradients. One gradient running from animal pole towards the vegetal pole, and the other gradient from vegetal pole towards the animal pole. Now, the animal gradient is more active at the animal pole region. Okay, there is a clear separation of animal pole and vegetal pole, and there is a clear separation of the metabolic activity rate as well. But you can differentiate these metabolic activities into two types: the animal pole metabolic activities and the vegetal pole metabolic activities. So now, animal pole metabolic gradient is very active in this region of animal pole and gradually decreases towards the vegetal pole. And the vegetal gradient is more active in vegetal pole and its intensity decreases when it moves towards the animal pole. Fine, I hope you can comprehend this. Okay, there is a clear separation of two gradients. Earlier they believed that there is only one gradient of metabolic activity. So one metabolic gradient which is moving from the animal pole towards the vegetal pole. That means the animal pole is more active and vegetal pole is less active. But later on, the modified theory clearly demarcated two gradients. The first gradient is the animal gradient, animal pole gradient. The other one is vegetal gradient. Animal gradient is more active towards the animal pole, and when it moves towards the vegetal pole, its intensity decreases. And the vegetal gradient is most active at the vegetal pole and its intensity decreases when it moves towards the animal pole. At the animal pole, the power to form structures, characteristics of this region are most pronounced. So how to differentiate between animal gradient and vegetal gradient? This is how you can differentiate. See, in the animal pole, the power to form structures, characteristics of this region are most pronounced. Okay. Similarly, at the vegetal pole, the power to form structures like archenteron, mesenchyme is greatest. Okay. In animal pole, certain structures like ectoderm, endoderm and other things will be more pronounced, but in vegetal pole, structures like archenteron, mesenchyme and other form structure, their power to form such structures is greatest in the towards the vegetal pole. As both gradients extend along the animal vegetal axis, they overlap each other and differ quantitatively. The animal gradient is believed to be cortical in location, while the vegetal is located more internally. See, you can see here the animal pole is very thin in nature, hence the animal gradient is situated more towards the periphery, that means towards the cortex, while the vegetal pole, you can see it is having more thicker cells, more larger cells, hence the vegetal gradient is situated more towards the center, medulla of the region rather than the cortex. But later work has shown that the gradients are due to the distribution of certain chemical substances like tryptophan and other nucleotides. The animal gradient promotes the protein synthesis while the vegetal gradient inhibits this process, so that protein synthesis may occur at different times along the animal and vegetal axis. The next and very important as well as a remarkable theory in the field of developmental biology is the organizer theory by Spearman. Spearman performed experiments on amphibian eggs during 1901 to 1912 with his student Carl Mangold, and observed that the formation of lens of eye in grass frog embryo is dependent upon the presence of optic vesicle. When the optic vesicle was transplanted into the belly region, the overlapping ectoderm differentiated into lens. See, while studying, he designed certain experiments which are now called transplantation experiment, where a part of a embryo is picked and transplanted in some other parts. Now what did he do is, he removed a part of grass frog embryo which is going to become the lens of the eye in the future, and he transplanted it on the belly region. What happened then? The overlaying ectoderm, that means the ectoderm present in the belly region, was later differentiated into lens. It became lens. So the removal of presumptive eye rudiment resulted in failure of lens formation. So the place where the lens was supposed to be formed did not form because he has removed the optic cup from there and he introduced it on the belly region. So eye formed in belly rather than in the actual region where it was supposed to be. In 1918, he transplanted the dorsal lip of the blastopore. You can see here, this is the blastopore, this is the gastrula of the frog, and there is an opening called blastopore, and just above the blastopore there is a place called dorsal lip. The dorsal lip of blastopore was removed from one of the frog embryo, and he transplanted it into the embryo of the another frog on the region which is supposed to become the ventral surface in the future. Okay, this place. It's the second embryo's dorsal lip is here. This is called primary dorsal blastopore or primary dorsal lip, and here the implantation took place the donor gastrula site. So here he implanted the second piece. That means he removed the piece from here and implanted a piece from this embryo. So you can clearly see the difference here, the red gastrula and yellow color, orange colored one, pale orange colored. So a part from here is removed and it is transplanted here. Now what happened? The dorsal lip, in the new site, the dorsal lip induced the formation of second embryo. A second embryo is formed. the dorsal lip induced the tissue surrounding it to differentiate into a new embryo or second embryo. Thus, some influence was passed on from the dorsal lip to other tissues, determining the development of the ectoderm of the gastrula. It was the cauda mesoderm situated in the roof of the archenteron, which had an organizing effect on overlying tissues. Transplantation of the skin ectoderm or the cauda mesoderm, the neural plate area, resulted in the differentiation of skin ectoderm into neural tissue. On the other hand, if a piece of neural ectoderm is transplanted in skin area, it lost its identity and differentiated into skin ectoderm. So this is how different potencies of different parts of the embryo was studied. Okay, if you take a part of upper lip of the blastopore, the dorsal lip of blastopore here, remove from here, introduce somewhere else, so it will now influence all the residing cell to become a secondary embryo. This is the normal embryo which are supposed to develop, and this is the secondary embryo. Because secondary invagination begins, the morphogenetic movements will begin, and ultimately a secondary smaller embryo is formed. A twinning is formed here. Same way, he also included many transplantation experiments like skin ectoderm or the cauda mesoderm and neural plate area. So now what happened? That particular area became skin ectoderm into neural tissue. Fine. That means the place was supposed to become skin in the future. Now it become neural tissue. That means nervous system. So if you press a neural ectoderm into the skin area, you implant a neural ectoderm into skin area. Now neural ectoderm lost its identity and it differentiated in skin ectoderm. Like that. If you transplant a particular part of the neuroectoderm to the skin area, then skin becomes the neurons, and if you transfer skin to the neuroectodermal area, and neurons become the skin. So this is the results of the transplantation experiments. So after this breakthrough, many workers subsequently reported the organizing influence of cauda mesoderm of many other animals, and efforts were made to identify and isolate the chemicals responsible for this inducing effect, about which in detail we are going to study in the coming sessions. Okay. Different workers reported different substances. Waddington and his co-workers noted that even a chemical or dye could function as an inducer. So externally, if you use some chemicals or dyes, that may also induce the embryo to develop into different structures and organs. So for his landmark discovery of dorsal lip of amphibian blastula acting as a primary organizer, Spemann received Nobel Prize in 1935. Unfortunately, his student who was actively involved in this research, Mangold, did not receive the Nobel Prize because 10 years before she died due to an accident in her kitchen while she was preparing milk to her newborn child. Organizers at present are recognized as an embryonic tissue that influence and organize the other tissues to differentiate and produce a tissue or structure that in normal course should not have been formed. This process is known as induction, and the tissue producing this effect as the inductor or organizer. So in detail we are going to study about this embryonic induction and about the inductor or organizer in the coming sessions and subsequent sessions of the same chapter. Thank you. For any further information about the theories of development, I would recommend you to follow these textbooks. Thank you.